Welcome, everyone. My name is Kara Burke, a pharmacist and team leader in the Division of Drug Information. I would like to welcome you to this educational activity sponsored by the FDA Center for Drug Evaluation and Research, Office of Communications, Division of Drug Information. Today's webinar is titled FDA Drug Topics, Regulatory Framework for Human Drug Compounding. Before I introduce our speaker, I just have a few housekeeping remarks. All faculty are expected to use generic names. If trade names are used, those of several companies should be used rather than only that of a single supporting company. Unapproved use disclosure. CE faculty and speakers are required to disclose to the attendees when products or procedures being discussed are off-label, unlabeled, not FDA approved, and any limitations on the information that is presented. Disclosure. The faculty's planning committee members and the FDA CE consultation and accreditation team have nothing to disclose. All inquiries for information relating to our webinars should be directed to ddIwebinars@fda.hhs.gov. We hope you'll enjoy meeting our presenters today. Marie-Stella Bouhet, JD, is a regulatory counsel in the Division of Compounding Policy and Outreach in the Office of Compounding Quality and Compliance in FDA Center for Drug Evaluation and Research. She received a Juris Doctor with a health law concentration from the University of Maryland School of Law and a Bachelor of Arts in Political Science from Barnard College, Columbia University. Prior to joining FDA, Marie-Stella worked as a senior litigation paralegal at Paul, Weiss, Rifkin, Wharton, and Garrison LLP in New York City. Dr. Madeline Wolfert is a clinical reviewer on the Pharmacy Compounding Review Team in the Office of Specialty Medicine in the Office of New Drugs and FDA's Center for Drug Evaluation and Research. She's a board-certified pediatrician who received her medical degree from Georgetown University and completed her pediatrics residency at Children's National Medical Center. Prior to joining FDA, she was a practicing pediatrician in a community outpatient clinic in Northern Virginia. Now, please give a warm online welcome to Marie-Stella Bouhet and Dr. Madeline Wolfert.
Thank you, Kara. Good afternoon, everyone. My name is Marie-Stella Bouhet, and I am a regulatory counsel in the Office of Compounding Quality and Compliance. I will pause here for a moment so that my co-presenter can briefly introduce herself as well.
Good afternoon. My name is Madeline Wolfert, and I am a clinical reviewer with the Pharmacy Compounding Review Team in the Office of Specialty Medicine, Office of New Drugs.
Great, thanks. Okay, let's begin. This presentation reflects the views of the authors and should not be construed to represent FDA's views or policies. Neither presenter today have anything to disclose. The first half of this presentation will provide a general background, history, and discussion on the regulatory framework on human drug compounding. The second half of this presentation will provide an overview of the evaluation process for bulk drug substances nominated for use in compounding in accordance with Sections 503A and 503B of the Federal Food, Drug, and Cosmetic Act, and how the evaluation process differs from the new drug approval process. In particular, this presentation will describe the history and regulatory framework of human drug compounding and the differences between Sections 503A and 503B of the FD&C Act. Madeline will also explain the evaluation process for bulk drug substances nominated for use in compounding in accordance with Section 503A of the FD&C Act and the 503A bulks list, and bulk drug substances nominated for use in compounding in accordance with Section 503B of the FD&C Act and the 503B bulks list, and how the evaluation process differs from the new drug approval process. The basics. Human drug compounding is generally regarded as the practice where a licensed pharmacist, a licensed physician, or in the case of an outsourcing facility, a person under the supervision of a licensed pharmacist combines, mixes, or alters ingredients of a drug to create a medication tailored to the needs of an individual patient. Compounded drugs play an important role for patients whose clinical needs cannot be met by an FDA-approved drug. However, compounded drugs are not FDA-approved. This means that FDA does not review these drugs to evaluate their safety, effectiveness, or quality before they reach the patient. As a result, they pose a higher risk than FDA-approved drug products. The Federal Food, Drug, and Cosmetic Act under Sections 503A and 503B include provisions for compounded drug products to be exempt from certain requirements of the Act if certain conditions are met. These will be discussed in more depth later in the presentation. During the 1990s, there was a significant growth in the number of pharmacies compounding drugs. By 1992, the agency became aware that pharmacies were engaging in activities consistent with conventional drug manufacturers. Instead of providing drugs to individual patients, pharmacies were supplying large quantities of drugs to hospitals and other customers nationwide. FDA also began to learn of serious adverse events associated with compounded drugs. In response, FDA published a compliance policy guide in 1992, which outlined FDA's enforcement policy on pharmacy compounding. After FDA published the 1992 CPG, the industry lobbied Congress, which resulted in several bills being introduced designed to limit the agency's oversight of compounding. In 1997, Congress enacted the Food and Drug Administration Modernization Act, or FDAMA, which added Section 503A to the FD&C Act. Section 503A describes the conditions that must be met for drug products compounded by a licensed pharmacist in a state licensed or federal facility, or by a licensed physician to qualify from exemptions from three key provisions of the FD&C Act: the new drug approval requirements in Section 505, the labeling with adequate directions for use in Section 502 F1, and the current good manufacturing practice or CGMP requirements under Section 501 A2B. To qualify for these exemptions, compounded drugs must meet the conditions set forth in Section 503A. We will go over these conditions in a few minutes. The Congressional Committee of Conference issued a joint statement explaining that it was the intent of the conferees in enacting Section 503A to ensure continued availability of compounded drug products as a component of individualized therapy, while limiting the scope of compounding so as to prevent manufacturing under the guise of compounding. As originally enacted, Section 503A included a prohibition on soliciting for prescriptions and advertising or promoting specific compounded drugs. A group of compounding pharmacies challenged these restrictions, arguing that these restrictions violated the First Amendment's free speech guarantee. Ultimately, the U.S. Supreme Court held in 2002 that those advertising prohibitions amounted to an unconstitutional restriction on commercial speech. The Supreme Court did not address the severability of that provision from the rest of the law. Because district courts held different opinions on severability, there was a lack of clarity on the law's enforceability nationwide. Despite the ruling, FDA was still able to investigate serious adverse events associated with compounded drugs. I'll point out a few on this slide. In 2007, 3 people died from multiple organ failure after a pharmacy called Apothecure sold super potent colchicine that was as much as 640% the labeled strength. In 2011, Meds 4 pharmacies' contaminated total parenteral nutrition products, or TPN, resulted in 19 cases of bacterial infection and 9 patients died. And in 2012, 43 patients developed fungal eye infections after receiving injections of contaminated sterile ophthalmic drug products compounded by Frank's lab. At least 29 of these patients suffered vision loss. In these cases, the companies recalled its compounded products. In October 2012, the United States faced the most serious outbreak associated with contaminated compounded drugs in recent history. You might be aware of the National Fungal Meningitis outbreak caused by the New England Compounding Center. NECC was a pharmacy located in Massachusetts that compounded vials of preservative-free methylprednisolone, which is a steroid. They shipped the steroid to patients and healthcare providers throughout the country. These drugs, which were contaminated with fungal growth, were injected into patients' spines and joints. The image to the right shows fungal growth in a patient's spinal fluid sample following the injection of the contaminated compounded product into the patient's spinal column. Approximately 14,000 patients received injections from the lots of contaminated drug product, and more than 750 people developed fungal infections, and more than 60 people died. When investigators inspected NECC, they observed greenish-black particles upon visual inspection of intact vials. Upon microscopic examination of the particles, it showed that there were masses of fungal hyphae. They also noted filthy and sanitary conditions at multiple locations within the facility. These and other violations contributed to the contamination of the compounded products. After the NECC outbreak, FDA engaged with members of Congress and other stakeholders in a bipartisan effort to pass legislation to prevent another outbreak. In 2013, Congress passed the Drug Quality and Security Act, which removed the unconstitutional advertising and promotion provisions of Section 503A that I spoke about earlier. The passage of the DQSA established Section 503A's validity nationwide. DQSA also added Section 503B to the FD&C Act, which added a new category of compounders called outsourcing facilities. This was to fulfill the need for supplies of non-patient specific compounded drugs held in physicians' offices and hospitals, or what we refer to as office stock, to treat larger numbers of patients who present needs that cannot be met by an FDA-approved drug. Section 503B provided for the first time a sector of compounders under FDA's primary jurisdiction and direct oversight, and these compounders are required to prepare drug products under CGMP quality standards, the same standards applicable to conventional drug manufacturers. I'll discuss this a bit more in the next few slides. I will now provide a brief overview of Section 503A and Section 503B and highlight some differences between the two. Section 503A describes the conditions that must be met for drug products compounded by a licensed pharmacist in a state licensed or federal facility or by a licensed physician to qualify for exemptions from three key provisions of the FD&C Act. They can qualify for exemptions from the new drug approval requirements under Section 505, the labeling with adequate directions for use under Section 502F1, and the current good manufacturing practice or CGMP requirements under Section 501A2B. Section 503B describes the conditions that must be met for drug products compounded in an outsourcing facility by or under the direct supervision of a licensed pharmacist to qualify for three key exemptions from the FD&C Act. These are 1, the new drug approval requirements under Section 505, 2, the labeling with adequate directions for use under Section 502F1, which are the same as under Section 503A, although Section 503B has other labeling requirements, and 3, the exemptions from the drug supply chain security requirements under Section 582. It's important to note that outsourcing facilities remain subject to CGMP requirements. What happens if a pharmacy or an outsourcing facility fails to qualify under Section 503A or 503B for the exemptions? The answer is if they fail to meet the conditions, the drug products compounded will not meet the exceptions, and that then means that all of the FD&C Act requirements applicable to drugs made by conventional manufacturers will apply. What are some of the limits on compounding? Many of the conditions in sections 503A and 503B are similar. For example, for both 503A and 503B, there is a prohibition on compounding drug substances that have been withdrawn or removed from the market because FDA has found them to be unsafe or not effective. Under sections 503A and 503B, there's also a prohibition on compounding drugs that are on the demonstrably difficult to compound lists that FDA will establish. Under Section 503A and 503B, there's also a prohibition from making what are called essentially copies of drugs. Under 503A, pharmacies cannot compound regularly or inordinate amounts of drug products that are essentially copies of a commercially available drug product. Under Section 503B, the essentially copy provision states that compounders cannot compound essentially a copy of one or more FDA-approved drugs. Other conditions under sections 503A and 503B are more distinct. One of the most important differences is that section 503A requires that all compounding be based on the receipt of a valid prescription for an identified individual patient. Unlike section 503A compounders, outsourcing facilities are not required to obtain a prescription for an individual patient. That means that outsourcing facilities are permitted to compound office stock. Section 503A also directs FDA to develop a standard memorandum of understanding, or MOU, for use by the states that addresses interstate distributions of compounded human drug products. There is no MOU requirement under Section 503B. In addition, there is a prohibition on wholesaling. This provision states that drugs may not be sold or transferred by an entity other than the outsourcing facility that compounded the drug, but there are some significant exceptions to this. In June 2023, FDA announced the availability of a draft guidance entitled Prohibition on Wholesaling under Section 503B of the Federal Food, Drug, and Cosmetic Act that, if finalized, will describe how FDA intends to apply Section 503B's wholesaling provision to outsourcing facilities that compound drugs. The statutory prohibition on wholesaling helps to ensure that compounding is based on patient need, which in turn reduces the overall risk of patient harm and helps to preserve the integrity of the U.S. drug approval process and of the U.S. drug supply chain. Another distinction here is that Section 503A directs FDA to create a list of bulk drug substances that can be used in compounding. Section 503B places more significant limitations on compounding from bulk drug substances. We will go over these differences in the next few slides. Let's look at who compounders under Section 503A are. They are state licensed pharmacies, federal facilities, and physicians authorized to compound. They number in the many thousands. They generally do not register with FDA. The pharmacies are primarily regulated by states. The frequency and depth of state oversight of the pharmacies varies from state to state. All 503A compounders must obtain prescriptions for identified individual patients. Compounding physicians are generally not routinely overseen by any regulatory body. An outsourcing facility is defined under 503B as a facility that is engaged in the compounding of sterile drugs, has elected to register as an outsourcing facility, and complies with all of the requirements found under Section 503B. We have a list of those registered facilities on FDA's website. Other points about 503B compounders. They are not required to be a state licensed pharmacy, but any compounding they do must be by or under the direct supervision of a licensed pharmacist. They are subject to increased federal oversight through FDA inspection. They are subject to current good manufacturing practice requirements. They are required to report adverse events to FDA. They are prohibited from wholesaling with exceptions of administration in a healthcare setting and dispensing. They are required to submit product reports to FDA biannually in June and December. And they are also required to label their products as directed under the Act. Let's talk in a bit more detail about compounding with bulk drug substances. Sections 503A and 503B define a bulk drug substance the same as an active pharmaceutical ingredient as defined in 21 CFR 207.1, and an active pharmaceutical ingredient is defined as any substance that is intended for incorporation into a finished drug product and is intended to furnish pharmacological activity or other direct effect in diagnosis, cure, mitigation, treatment, or prevention of disease, or to affect the structure or any function of the body. But the term does not include intermediates used in synthesis of the substance. Active pharmaceutical ingredients do not include excipients or inactive ingredients. Under Section 503A, if a pharmacy is compounding with a BDS, that substance must comply with an applicable United States Pharmacopoeia or USP or National Formulary or NF monograph, if one exists, and the relevant USP chapter on pharmacy compounding. Or if an applicable USP or NF monograph does not exist, the BDS can be a component of an FDA-approved drug. This means that under Section 503A, pharmacies and physicians can compound with a BDS that has an applicable monograph or is a component of an approved drug as a matter of law. If a monograph does not exist and the substance is not a component of an FDA-approved drug, it must appear on a list of bulk drug substances that can be used in compounding under Section 503A, developed by FDA through regulations. Under Section 503B, there are more significant limitations placed on compounding from a BDS. If the facility is compounding from a BDS, the BDS must appear on a list of bulks for which there is a clinical need, or the drug compounded from the BDS must appear on FDA's drug shortage list at the time of compounding, distribution, and dispensing. Also, under Sections 503A and 503B, the BDS must be accompanied by a valid certificate of analysis and must have been manufactured by an establishment registered with FDA. While the bulks lists are in development, FDA currently has interim policy guidances under Sections 503A and 503B that categorizes bulk substances while FDA evaluates them. Category 1 bulk substances are substances currently under evaluation and were nominated with sufficient supporting information and do not appear on any other list. Category 2 substances are substances that raise significant safety concerns. And Category 3 substances are substances that were nominated without adequate support. The interim policy guidance under Section 503A describes the conditions under which FDA does not intend to take action against a licensed pharmacist or licensed physician for compounding a drug product from a bulk drug substance that is not the subject of an applicable USP or NF monograph, is not a component of an FDA-approved drug, or does not appear on the list of bulk drug substances that can be used in compounding under Section 503A of the FD&C Act while FDA is developing the 503A bulks list if certain conditions are met. The interim policy guidance under Section 503B describes the conditions under which FDA does not intend to take action against an outsourcing facility for compounding a drug product from a bulk drug substance that does not appear on a list of bulk drug substances that can be used in compounding and is not used to compound a drug product that appears on the FDA drug shortage list at the time of compounding, distribution, and dispensing, while FDA develops the list of bulk drug substances that can be used in compounding pursuant to Section 503B of the FD&C Act, while FDA is developing the 503B bulks list if certain conditions are met. Nominations for the 503A bulks list may be submitted to an open FDA docket, and there is an associated Federal Register notice that provides instructions on the information that should be submitted, along with the name of the BDS, to enable FDA to have enough information to evaluate the nomination. FDA must develop the 503A bulks list through regulations and in consultation with the United States Pharmacopoeia Convention, or USP, and the Pharmacy Compounding Advisory Committee, or PCAC, under Section 503AC. FDA has been evaluating substances for inclusion on the 503A bulks list on a rolling basis and publishes notices in the Federal Register to communicate its intentions and decisions regarding groups of substances that have been nominated. Substances nominated for the 503A bulks list with adequate support are evaluated by FDA and presented to our Pharmacy Compounding Advisory Committee, which makes a recommendation on whether to add the substance to the list. Then FDA publishes a proposed rule in the Federal Register, followed by a final rule in the Federal Register, either adding or not adding the substance to the list. The 503A bulks list is codified at 21 CFR 216.23. Currently, there are 6 bulk drug substances on the 503A bulks list, and they are listed here for your reference. Let's talk now about how FDA develops the 503B bulks list. One of the conditions under Section 503B is that the drug is compounded in an outsourcing facility that does not compound using a bulk drug substance unless the bulk drug substance appears on the list identifying the bulk drug substance for which there is a clinical need, or the drug compounded from the bulk drug substance appears on the FDA drug shortage list at the time of compounding, distribution, and dispensing. FDA published a final guidance in March 2019 to explain how we intend to evaluate whether there is a clinical need to compound with a bulk drug substance under Section 503B. The guidance states that the outsourcing facility must show that there is a clinical need for an outsourcing facility to compound the drug product. For example, the clinical need cannot be met by an existing approved drug. The outsourcing facility must also show that the drug product must be compounded using the bulk drug substance and that it is not feasible for them to compound from an approved drug. Nominations for the 503B bulks list are received in a similar way to those for the 503A bulks list. FDA maintains a docket for nominations that is open, and an accompanying Federal Register notice provides additional information on what should be submitted as part of a nomination to enable us to evaluate that particular bulk drug substance for inclusion on the list. Similar to 503A, FDA evaluates these substances on a rolling basis, and we periodically publish notices in the Federal Register to communicate intentions and decisions regarding groups of substances. The substances nominated for the 503B bulks list with adequate support are evaluated by FDA. Unlike Section 503A, FDA is not required to develop the 503B bulks list via rulemaking, but instead directs the FDA to publish notices in the Federal Register proposing and then designating bulk drug substances for inclusion on the 503B bulks list. The 503B bulks list is housed on FDA's website. Currently, there are 5 bulk drug substances on the 503B bulks list, and they are listed here for your reference. I will now pass the presentation on to Madeline, who will go over the evaluation process for bulk drug substances and provide specific examples.
Thank you so much, Maria Stella, for leading us through that information about the regulatory framework and the 503A and 503B list. Again, my name is Madeline Wolfert, and I'm with the Pharmacy Compounding Review Team. Our team works collaboratively with Maria Stella's office, the Office of Compounding Quality and Compliance. And now that she has reviewed the regulatory components of compounding, I will discuss some of the clinical aspects and delve into how FDA evaluates these substances under 503A and 503B. Today, I'll focus on the 503A evaluation process, which again would be compounding for individualized therapy or patient-specific prescriptions. For substances nominated for the 503A list, we evaluate information provided by the nominator to support their nomination. Ideally, that would include information like pharmacokinetic data and clinical data. We also then search for publicly available information from published medical literature, clinical guidelines, various databases, and websites. This is in contrast to new drug applications, where a drug company or sponsor submits data, which is often proprietary. FDA assesses each substance according to 4 criteria: physical and chemical characterization, safety issues raised by use in compounded products, evidence of effectiveness or lack of effectiveness, and historical use in compounded products. No one criteria is dispositive, and we balance them on a substance-by-substance basis. We will provide examples of substances evaluated under 503A later in the presentation. Again, this is our process evaluating substances for the 503A bulks list. For 503B, we use similar 4 criteria to evaluate whether there is a clinical need for the compounded substance, in addition to considering whether the nominated substance is a component of an approved product or not. More information on how FDA evaluates substances for the 503B list can be found in the FDA guidance documents and resources listed at the end of this presentation. Before I go through examples of recent substances that we've evaluated for the 503A list, I wanted to give you a brief, high-level overview of differences between the new drug application review process and the compounding evaluation process. Here we've listed some of the differences between these 2 processes, but please note the differences are not limited to this list. First, under the NDA process, an application is submitted to FDA by a sponsor. In compounding, a nomination of a bulk drug substance is submitted to FDA by a nominator. Note that, as Maria Stella previously described, nominations and evaluations for the 503A and 503B lists are done separately. Second, NDA sponsors submit data and are required to demonstrate substantial evidence of effectiveness and show that the drug is safe for the intended use. Clinical data are usually from adequate and well-controlled studies. For compounding, the nominator submits publicly available evidence of effectiveness and safety information to support the nomination. Nominations must include a bibliography of safety and efficacy information, including any relevant peer-reviewed medical literature. But note that the data may not be of the same caliber, type, amount, or quality as is required to support an NDA. Third, NDA sponsors apply for a specific indication. For compounding, The nominator provides a proposed use or uses for the compounded drug product, basically a difference in terminology here. And fourth, for NDAs, FDA reviews CMC information and final product formulation. That is, the finished dosage form containing active and any inactive ingredients. In compounding, FDA evaluates the active pharmaceutical ingredient or drug substance, not specific drug product formulations or manufacturing processes. Next is on labeling. For NDAs, drug product labeling must meet content and format requirements. Compounded drugs are exempt from labeling of drugs with adequate directions for use. However, for drugs compounded under 503B, the label and container must contain certain information, such as the statement, "This is a compounded drug." Next, for NDAs, FDA generally performs pre-market assessment, for example, facility inspection, of the establishments where the drug product will be manufactured. For compounding, FDA conducts surveillance, for-cause, and follow-up inspections of compounders, or inspects according to a risk-based schedule for 503B facilities, taking into account, for example, the inspection frequency and history of the outsourcing facility. Inspection is not drug-specific. And finally, once an NDA is approved, the sponsor can make that claim that a drug is FDA approved. Compounded drugs are not FDA approved and should not be equated with FDA approval, endorsement, or recommendation. Now we will share 2 examples of substances that were recently evaluated for the 503A list to demonstrate the process. Our first example is melatonin. Melatonin was nominated for the 503A list for oral use for the treatment of sleep disorders in children and adolescents with autism spectrum disorder, ASD. There are no FDA-approved products for the treatment of insomnia in children and adolescents. As quick background information, melatonin is a neurohormone that plays a key role in regulating the sleep-wake circadian rhythm. Sleep disorders tend to be more common and severe in children with neurodevelopmental disorders like ASD. Between 44 to 83% of children with ASD are reported to experience sleep disorders, and these coexisting sleep disorders can have significant effects on patient health and quality of life, family functioning, and can also worsen the core symptoms of ASD. So let's go through the evaluation of melatonin. FDA evaluated melatonin using the 4 criteria that I described previously. For the assessment of physical and chemical characterization, FDA concluded melatonin is easily characterized and preparation of melatonin has been well developed. For safety, based on both non-clinical and clinical information, melatonin was found to be relatively safe and without serious adverse events. For effectiveness, clinical trials showed a treatment response in short-term treatment of sleep disorders in children and adolescents with ASD. We also reviewed clinical practice and professional society guidelines, some of which recommend melatonin in the population of patients with ASD. The EMA, European Medicines Agency, has also licensed a prolonged release dosage form of melatonin in ASD. And for the final criterion, historical use, we found that melatonin has been used in compounding since at least 2009 in a variety of dosage forms. It's approved for use in Europe, Australia, and Japan. In June of 2021, melatonin was presented and discussed at the Compounding Advisory Committee, or PCAC meeting. FDA made the recommendation after balancing the 4 criteria that weighed in favor of adding melatonin to the list. The advisory committee voted, concurring with FDA, to include melatonin on the 503A bulks list. And it will now go through the rulemaking process, proposed then final rule that Mary Estella described earlier. The second example we'll discuss is oxytryptan or 5-HTP. Oxytryptan, which is also known as 5-hydroxytryptophan or 5-HTP, is a chemical precursor in the biosynthesis of the neurotransmitter serotonin from tryptophan. In June 2015, oxytryptan was discussed at a compounding advisory committee meeting. FDA had evaluated oxytryptan for inclusion on the list using 4 criteria for the nominated conditions depression and sleep disorders. FDA did not recommend adding it to the list due to a lack of evidence of effectiveness, as well as safety concerns, including risk for serotonin syndrome and inadequately treated depression. The advisory committee voted not to include it on the 503A list, and taking into consideration the committee's advice, FDA determined not to include oxytryptan on the list. In February 2019, after the rulemaking process, the final rule was published in which oxytryptan was not included on the list. Thereafter, healthcare providers and caregivers of patients with BH4 deficiency contacted FDA expressing that oxytryptan is an essential treatment for pediatric patients with BH4 deficiency, which is a rare disease that we'll discuss further in the next few slides. After FDA issued guidance that it does not intend to take action against compounders who use oxytryptan for BH4 deficiency, FDA reevaluated oxytryptan in June of 2021 for use in BH4 deficiency and presented to the Compounding Advisory Committee. We'll talk about this evaluation in more detail. So what is BH4 deficiency? In the next slides, we'll discuss BH4 deficiency and its treatment. BH4 deficiency is a group of treatable genetic neurotransmitter disorders. Deficiency of BH4 can manifest with hyperphenylalaninemia or elevated blood phenylalanine levels and deficiency of oxytryptan and L-DOPA. Symptoms can include motor dysfunction, impaired muscle tone, movement abnormalities, intellectual disabilities, and seizures. Treatment should be initiated as early as possible because late detection and late initiation of effective treatment can lead to irreversible brain damage. And we'll discuss treatment on the next slide. The treatment strategy is two-pronged. First, a restricted phenylalanine diet and/or BH4 replacement. And secondly, in combination with phenylalanine control, substitution of depleted neurotransmitters, serotonin and dopamine, by using their precursors, such as oral oxytryptan. Oxytryptan is considered a first-line treatment according to professional organizations, and the dosing is based on weight and adjusted depending on clinical response. We evaluated oxytryptan for oral use in BH4 deficiency using the 4 criteria discussed previously. For physical and chemical characterization, we concluded oxytryptan is well characterized and likely to be stable in solid and solution formulations. For safety, available non-clinical data did not identify significant safety concerns. For clinical safety, the most common adverse events of oxytryptan in patients with BH4 deficiency include gastrointestinal symptoms, irritability, motor disorders, and sweating. There's also the potential risk of serotonin syndrome, especially if combined with another serotonergic agent. In terms of effectiveness, oxytryptan is considered a standard therapy in BH4 deficiency with numerous case reports. And finally, oxytryptan has been used in compounding since at least 2011 based on the literature. However, it can be presumed that it's been used since the 1970s when dosing for children with BH4 deficiency was first described. In June 2021, oxytryptan was presented at the Compounding Advisory Committee meeting for use in BH4 deficiency. FDA's recommendation was that a balancing of the 4 criteria weighed in favor of adding oral oxytryptan to the list. And the advisory committee also voted to include it on the 503A list. It will now go through the rulemaking process. So finally, we'd like to share some resources on human drug compounding. This slide lists 3 recent guidances for compounding under Section 503A and 503B. Note that some of these guidances are being revised. And finally, FDA's website on human drug compounding has very helpful information, including additional training, and language from sections 503A and 503B can also be found on the website. So this concludes our presentation. At this point, I'd like to thank Kara and the DDI team for having us today and helping to run today's webinar. I'd also like to thank all of our FDA colleagues who helped with this presentation and who are involved in the compounding program. And finally, I'd like to thank all of the participants for attending today's webinar. Thank you all so much for your attention.
All right. Okay. Once a substance is added to the 503A or 503B bulks list, can compounders claim that FDA considers it safe or effective?
Great, so I can take that question. So the short answer to that is no. Inclusion on the 503A or 503B bulks list should not in any way be equated with or considered an FDA approval or endorsement or recommendation of any drug compounded using the substance. Nor should it be assumed that a drug compounded using a substance included on the list has been proven to be safe and effective under the standards required to receive agency approval.
Okay, thank you so much. All right, let's do 2 more. The next question that came in, are melatonin and oxytryptan available as dietary supplements?
This is Madeline. Yes, thank you for your question. Yes, these are available as dietary supplements. However, these active pharmaceutical ingredients or bulk drug substances have also been nominated for use in compounding under Section 503A of the FD&C Act as prescription compounded drugs. So FDA has a separate process for evaluating bulk drug substances for the 503A bulks list. Availability of a product as an ingredient in a dietary supplement is not one of the criteria that FDA considers in its evaluation, and also inclusion or exclusion from the 503A bulks list does not affect their availability as dietary supplements. Additionally, FDA does not consider USP monographs for dietary supplements to be applicable USP or NF monographs within the meaning of Section 503A. Thank you.
Okay, thank you so much. Well, since we're after the 2 o'clock mark and I know others have to get to meetings and other obligations, let's do one more question. Right, how do I submit comments regarding FDA's policies?
Okay, I can take that question. So for instructions on how to submit comments concerning specific either draft or final policy documents that FDA has issued, folks should follow the instructions in the Federal Register notice announcing the availability of that particular document. You can search for specific policy documents and their notices of availability on www.federalregister.gov. You should also be aware that FDA has also established a public docket with docket number FDA 2015 N 0030 to receive information, recommendations, and comments on matters related to the agency's regulation of compounding of human drug products under sections 503A and 503B of the FD&C Act. This particular docket is intended for general comments related to human drug compounding that are not specific to documents or issues that are the subject of other dockets. Thank you.
Okay, thank you so much, Maricela and Madeline. That was a great presentation, very informational. I know we did have a lot of questions and we just ran over time. So if you had questions that we did not have time to address today, please email them to either the compounding division, which is compounding@fda.hhs.gov, or our Division of Drug Information email address at druginfo@fda.hhs.gov for assistance. And if you need more CEs for your license renewals this year, please check out our list of home study CE webinars at www.fda.gov/DDI webinars. And if you have your phones out, we have 5 QR codes in front of you on that one slide that is easy to access. And one is the continuing education. There's Twitter to subscribe to our listserv, our podcasts, and also our Gaddis LinkedIn group. So if you have your phones out, please scan them for even more information and CEs. You may also find additional free CEs such as FDA's drug regulation course titled FDA's Role in Public Health, Drug Efficacy, Safety, Quality, and Beyond on our CDER Learn website by entering CDER Learn in the search engine on fda.gov. This concludes our activity for today. The FDA appreciates your participation in this webinar, and we hope that you will join us again in the future. Thank you everyone for attending, and please continue to stay safe and healthy. Take care.